Okay, I think it's on, right? Okay, perfect. So, good afternoon, everyone. Thank you for having me here today. I learned that this is this is going to be the first talk in which we try this virtual session. So I hope everything goes okay, but I'm sure it will. And I'm very happy because I have this opportunity today to tell you something about what we do at the UK Centre for Astrobiology here at the University of Edinburgh. I am a postdoctoral research associate in Charles Cockell Group. And we are in JCMB, so in the School of Physics and Astronomy, but I am a microbiologist. And today I'm going to talk about the smallest astronauts, so just something about space microbiology in particular. And I hope that I convince you that microorganisms could be important in space for us, basically for us humans. So the content of the talk will be the following. First, I'm going to tell you something about what is a microorganism, because not necessarily everyone knows what we mean with that, and which are the roles of microorganisms on Earth. I'm going to focus on a role in particular, that is biomining, because it's important for, well, you will see why. Then I'm going to tell about the possible roles of microorganisms beyond Earth, so in space, and what about biomining in space in particular. And then I'm going to talk about BioRock. There is one space experiment that we performed on the International Space Station in last year, so in summer 2019, and then what to expect from the future. So first of all, what is a microorganism? Well, as I said before, not necessarily everyone is aware or is familiar with this term. So what you can see here is the evolutionary, it's actually a graphic representation of the evolutionary tree of life. That is a representation that biologists normally use to classify all the living organisms that we know. And if we look at that and we talk about microorganisms, we are referring to archaea, fungi, protists, and bacteria. And so, as you can see here, actually, microorganisms are quite diverse. They can be unicellular algae, they can be archaea, bacteria, unicellular fungi or filamentous fungi, viruses as well, and also protozoan. They have some characteristics in common, although, as you can see, they are quite different. The first is that they are very small. That's why they are called microorganisms. But the other two characteristics that they have in common is, the first, they are all single-cell organisms, or even less in the case of the viruses, for instance. They are also called unicellular organisms. And the second one is that they are everywhere. They've been found almost everywhere on Earth, everywhere we search for. And when I say everywhere, I literally mean everywhere, because they have been found on soil, of course, but also on several surfaces, like for instance in pipes, etc., but also in very hot environment and very cold environment, very dry environment, very salty ones, but also on clouds, and last but not least, also on plants and animals, including our own human body. In fact, it has been estimated that for every cell that composes our body, there is a bacterial cell or a microbial cell anyway. So I think how much we are composed of actually human cells and how much we are actually composed of microorganisms in some sense. And they are absolutely necessary for us because they allow us to be healthy. In fact, when we talk about microorganisms, we often think about something that is nasty, particularly in these unfortunate times in which a pandemic is going on. But more often, they are actually necessary for our own survival here on this planet. And in order to convince you about that, I want to show you some of the possible roles of microorganisms, that actually the roles that microorganisms do perform for us on Earth nowadays. One is, for instance, in the food industry, but also in some industries such as biofuel. They were absolutely important on early Earth for the composition of oxygen that we have nowadays, so eventually for our own respiration. But also they are used in industries, bioindustries such as pharmaceutical industries, but also biopolymers industries. They are important in the recycling of waste, in the production of fertile soil, also in bioremediation. So for environmental issues, bioremediation is basically the use of microorganisms to degrade pollutants from several environments. And last but not least, also on biomining. So what is biomining? Biomining is also called bioleaching. These two terms are used as synonyms, so this is what I'm going to do from now on. It is the process of using microorganisms to extract useful metals from rock ores and mine waste. It is actually quite widely used on Earth. In fact, 20–25% of copper is currently extracted by biomining. 5% of gold, but also nickel, cobalt, uranium, rare earth elements, and so on are nowadays extracted on Earth with biomining rather than with traditional mining. And there are some advantages of using biomining. That is, for instance, the fact that it's generally more economic and also more environmental friendly in general terms. So we saw how microorganisms are important on Earth. I want to show you now how they could be necessary for us in space. Actually, I correctly said how they will be absolutely necessary for us in space. In fact, some of the possible roles that they will perform for us is the generation of food, and this is strictly correlated to the production of fertile soil from extraterrestrial rocks and regolith. Also, on the generation of oxygen, we saw how important they were on Earth, on early Earth, so they will be important on an extraterrestrial planetary body. Also for the recycling of waste, and this is going to be extremely important when you think about the fact that a space station or an extraterrestrial settlement is going to be, at least in the beginning, an enclosed environment, so we need to recycle as much as possible. And eventually also in bioindustries, in advanced bioindustries. So all these roles actually go to support what we call life-sustaining self-support system, but also they are going to advance our capacity of performing in situ resource utilization. That basically is just the concept of thinking about using the resources in the place in which we are going to settle, rather than constantly relying on resupply from Earth, because it's not going to be sustainable. And if you think about it, the farthest we will go from Earth, the more this becomes not sustainable. So we need to rely on resources in the place in which we are going to settle. And from this point of view, space biomining, as you can imagine, is rising and increasing interest, because from the point of view of in situ resource utilization, it's going to be quite a thing, hopefully. And when we think about biomining in space, the idea is to go and biomine rocks and metals on the moon, on Mars, but also on asteroids. And this is going to be positive, of course, for a future space settlement, but some researchers also think that in future it may be good also for terrestrial industries. Nowadays it's not really sustainable, but maybe in future, who knows. It is with this idea in mind, the idea of try and test space biomining, that we decided to perform this space experiment called BioRock. It was launched, as I mentioned before, in summer 2019, but it was first proposed by my supervisor Charles Coquelle in 2018 to European Space Agency. So it took a long time to actually go in space. However, it wouldn't have been possible without the effort of former and present researchers and also students. And you can see here some of the companies and institutions that were involved in BioRock. So the motivation for BioRock was, of course, trying to understand if we could perform space biomining on some extraterrestrial material, and using regolith for in situ resource utilization, as well as for a future life support system, extraterrestrial life support system. But in order to understand that, we first needed to understand how microorganisms could actually, the biomining capacity of some microorganisms could have been affected by the space condition. One of the conditions there is in space is the lower gravity condition. That is also called microgravity, when we talk about, for instance, the condition in free space, on the International Space Station or so, but also on Moon and Mars, the condition is lower in respect to terrestrial gravity. So the first question to be addressed, if we wanted to understand something about space biomining, was try and understand how does gravity influence the way in which microbes interact with rocks and the mineral surfaces. Now I'm going to show you this video that was prepared by NASA before the experiment was actually launched. So you will notice that they speak in this video with the future tense, but this is just because it was prepared in preparation for the experiment itself. I hope you get the audio, otherwise just shout, unmute yourself and tell me.
The International Space Station will soon host some of the smallest miners in the universe, microbes. Microbes growing on the surface of rocks can gradually break them down and extract useful minerals and metals. This is a process called biomining. As we explore space, we are seeking to use biomining to turn rock and regolith into soil for growing plants and food. But before we can use this technique in planetary settlements, we first need to test it in space. On the space station, bioreactors will be placed inside a centrifuge where microbes will grow on rocks in microgravity and simulated Martian gravity. Investigators will examine how 3 types of microbes behave within pieces of basalt and evaluate how well the different microbes extract elements from the rocks. The findings will be compared to ground-based results. We hope to gain insights into how microbes interact with rocks in microgravity and how we might use them in our exploration of deep space.
Great. So I hope you heard that. I didn't hear anyone, so I guess the audio was okay. And so let's go a little bit into the details of BioRock. We first of all needed to select the right microorganisms to be used in this experiment. And these needed to have some characteristics in common, like for instance, the demonstrated capacity to grow on soil or rocks or to biomine, resist desiccation and others, but also needed to be safe for the crew on the space station. So eventually we decided for these 3 microorganisms here. One was Phingomonas dissecabilis that was provided by our group here at Edinburgh. The second one was Bacillus subtilis that was provided by the German Aerospace Center. And the third was Cupriavidus metallidurans provided by a Belgium center that is called SEK-CEN. And then we needed to select the rock substrate. We decided to go for basalt for 2 main reasons. The first is that basalt is a rock that is quite porous, and so it would have provided a good support for bacterial colonization. But the second is that basalt is actually a good analog for Martian and lunar rocks. So we selected this Icelandic specimen of rock, and we cut it, well, not necessarily this, actually this is a picture that I've taken online, and we cut it into pieces that were 13 millimeters each side, so quite small. And each basalt slide here was basically represented a single sample. Then we prepared the samples themselves. And how did we do that? We basically prepared a single bacterial culture, and we inoculated it on the basalt slide. So we had different basalt slides containing each one a single bacterial species, or also we have this that is called the non-biological control or negative control that was basically just a sterile basalt slide. So it was a control in which the bacterium was not, any bacterium was not present, basically. Once prepared, the samples were inserted into this hardware that is called biomining reactor, and that was specifically developed from Kaiser Italia and with a collaboration with our group for this experiment. And as you can see here, each hardware allocates 2 samples, and here you can see an hardware without samples. And you can see here a bunch of these hardwares ready for the launch, basically. So, okay, they were ready. You can see here some pictures of, basically the experiment was launched on the International Space Station in July 2019 from the NASA Kennedy Space Center in Florida. And we were lucky enough to be there for the preparation of the launch. And you can see here me, Charles, and Annemiek. There is a PhD student in our group. We were very happy here because we finished assembling all the hardware. And here you can see Charles handling all the hardware to the space station processing facility. And here part of the BioRock group that we were just going around the Space Center after working hard on this. So the launch was SpaceX Commercial Resupply Service 18, and here it's me on the launch site. And this is so we were lucky enough to actually attend the launch. And here you can see the astronaut Luca Parmitano installing the BioRock hardware into the cubic incubator on the ISS that you saw a representation of that in the video that I showed you before. And they stayed on the International Space Station for 1 month, more or less, and then they came back to us. Here it's Charles and me, very relieved that everything goes okay and that we had our samples back in our hands. And this is me analyzing the samples at the microscope. So let's go a little bit into the details of the experiment itself. We had, as you saw, 3 different bacterium and a negative control. So we prepared them. We prepared 4 different sets of samples. We included triplicates. So 3 sets of samples were launched on the International Space Station, and the 4th stayed on Earth. So for those that were in space were subjected to either microgravity, that is basically commonly referred to as absence of gravity or weightlessness. Simulated Martian gravity, that as you can see here is actually lower than terrestrial gravity because it's 0.4 g. And then simulated terrestrial gravity, that is 1 g. Then the 4th set of samples, the one that stayed on Earth, represented what we call the ground control. And in this case, only true terrestrial gravity was applied to these samples. So let's go into the results. What did we discover from BioRock? So the first thing that we analyzed, that we wanted to understand, was is the gravity condition able to somehow change the way and the final cell concentration of the bacterial species? The results, quite surprisingly, showed that there was no difference in the final cell concentration between the space samples after the 21 days in which the bacterium grew. And so what does this mean from our purpose? Basically, this is a typical, it's called a growth curve, and it is a typical way by which microorganisms grow. Because of how our experiment was designed, we didn't know, we had no idea what happens during this time, so during the 21 days, but we only had this last time point here, so after the 21 days. The fact that they all reach the same final cell concentration tells us that no matter what happens before the 21 days, after 21 days, they will all reach the same condition regardless of the gravity, the sun concentration, sorry, regardless of the gravity condition. And this is a good news from the point of view of biotechnological application, because it means that biotechnological application won't be negatively affected by the gravity conditions. And so this is the first actually result that we got from BioRock, and is that low gravity is not a limit to the biotechnological application in space. But I made a big deal about biomining, so what about biomining? Let's see what I got from this point of view. In order to address this question, I have to first introduce rare earth elements. These are a group of elements that are, despite the name, are actually quite abundant on Earth, and they are very important for our everyday life. They have an enormous impact on that because they are present in every single electronic device, even the simplest one. So considering how dependent we are nowadays on electronic devices, you can imagine how much we will be dependent on them in space. So where do we find rare earth elements? Well, as I say, they are abundant on Earth, but they are also found in specific rocks types called creep on Moon. They have been found on Martian meteorites, so we know that they are present on Mars as well, and they are also present on asteroids. So what did we discover? We analyzed which rare elements were bioleached or biomined by our microorganisms, and we discovered that one of the 3, that is actually Sphingomonas dissecabilis, so the one that was provided by our group, was able to extract rare earth elements in all the 4 gravity conditions that we tested. So very good news, because this means that space biomining is in principle achievable under a range of gravity conditions. And this also makes BioRock the first experiment that actually demonstrated biomining on a space station. So as a summary of the results that I showed you so far, we are still analyzing some other samples coming from BioRock. So hopefully in some months I'm going to give you other results. But from what we know so far from BioRock, we provided the first demonstration of biomining on a space station, and also the fact that biomining is in principle achievable under a wide range of gravity conditions. We also show that the final bacterial population are not negatively affected by the gravity condition, and this is a good news from the point of view of space by industrial application. We also demonstrated the functionality of a novel space biomining reactor, and this may be of some interest for those of you that are working in engineering, for instance. We discovered also a novel biomining microorganism, that is Sphingomonas dissecabilis. So Sphingomonas is actually already, as an organism itself, it was already known, but we didn't know that it could biomine. And this is actually a good example of how space studies can actually benefit also terrestrial applications, because now we can think about using that for terrestrial biomining, for instance. Because of the success of BioRock, we very recently launched a second space experiment called Bioasteroid that has a very similar scheme to BioRock, but it's going to be a little smaller. In fact, in this case, the only PI group that is going to be involved is our group at the UK Centre for Astrobiology. And the main difference is that this time we are going to biomine real extraterrestrial rock, because we are going to mine asteroids. And we are going to use Sphingomonas dissecabilis again, because it behaves so well for BioRock. But this time we are also going to use a different type of microorganisms, that is a filamentous fungus, Penicillium simplicissimum, and a mix of the two. And I'm very glad to tell you now that the experiment was successfully launched last Sunday on this mission, SpaceX Commercial Resupply Service 21. Unfortunately, this time, because of the COVID, we couldn't go in Florida to watch the launch. So this is me observing the launch virtually, but it was still exciting. And so let's conclude with what can we expect from the future. So there is a lot yet to be understood. For instance, with Bioasteroid, we hope that we can increase our knowledge of space biomining, but also on bioleaching and also rock weathering in space. That is another important topic that we addressed with BioRock. And we need to learn more about the bioleaching mechanism of Sphingomonas dissecabilis, and we are actually working on this right now. We need to understand more about the effects of gravity, because it can depend on the microorganism, of course, but also on the application. And we also need to select the best microorganism for a given space application that you want to do. We need to understand how to scale up the system and how to develop technologies to do that, depending on the application, of course, and also how they are going to be affected by long duration of space flights, how microorganisms are going to be affected, and also how to keep the process sustainable. And this is something I'm really keen into underlying, basically. So research is needed from this point of view, and our group is always open to collaboration. So if you have any bright idea, or you're interested, or you want to know more, just drop an email to me or Charles. And I want to stress again that this type of research is going to benefit space science, of course, but also the terrestrial application, as demonstrated by the discovery of a novel biomining microorganism. So I conclude now. I think I've taken a little more time than expected, so sorry about that. I want to thank all the excellent BioRock team. As you can see here, it was quite a big group. And I want to thank you for your attention, and I will take your questions. But if you don't want to ask me anything here, you feel free to contact me by these contacts here on the left. So thank you.